Welcome to the Astrobiology Radiation application, the Astro Rad-Bio-App. You can select from a range of different dashboards that allow you to examine radiation data related to spaceflight. There's a few different ways to select these different tabs, and there's metadata available to help you select the one of interest. The menu here is a great introductory starting point. You can see we can look at the average dose rate, the terrestrial dose rate, and then compare to lunar and Martian radiation doses. There's a glossary of terms at the end, so if you encounter something you don't recognize, you can look it up. You can filter based on the species and analysis method, or the type of organism you're using, the radiation type, or the general radiation groups that exist based on how the data was collected, be it on the International Space Station, US lab, or other spaceflight missions, or terrestrial experiments designed to investigate the effects of ionizing radiation on Earth. The table on the left allows you to access these data. You can examine them and understand them a bit more. It allows you to see some of the associated metadata. You can download this by pressing on the green arrow, and this JMP button appears, allowing you to download the data as XLS or Word. Save that for a later date. Can go to the next dashboard along. This allows us to examine the terrestrial radiation more closely. I'm just going to filter down to look at these lower dose experiments. These particular studies have extra metadata associated with them, such as the amount of energy in the photons or the dose rate during the experiment. Some experiments allow us to calculate the total dose, and we also have recorded the particle let for a subset of the studies. Here we can examine and compare the spaceflight studies to these terrestrial studies. We can filter and say just look at the spaceflight studies. Here we can see that the longer you spend in orbit, the exponential duration increases, and the absorbed radiation dose also increases. There's a correlation between the two. You can explore the data, and you can download it from this table on the left. I'm going to remove these filters and then go to the next dashboard. So this is the lunar crater radiation measurement lunar orbiter. This allows us to see 10 years worth of space weather data. And here in the next tab, you can see we compared these data to these terrestrial experiments that have been curated by the GeneLab team. Within this dataset, we have included the Martian transit data and the Martian surface data provided by the Curiosity rover team. And so now we can select a subset of the GeneLab accessions, which were exposed to similar amounts of radiation to that experienced en route to Mars and on the Martian surface. So this subset of studies we can now investigate at a further level using the GeneLab processed genetic data and try and see if there's any conserved genetic patterns that might help inform us about potential consequences of radiation exposure en route to Mars and on the Martian surface. Once more, these data, once you've filtered down to them, can be exported from the table on the left for further analysis. I recommend taking the list of accession numbers of GeneLab studies from that table and then using the GeoLab metadata exploration app to filter through and find the related GeneLab omics data, be it transcriptomics, proteomics, or metabolomics, as that provide more insights. At the end of the app, we've just included a link to a conversion tool that allows you to convert between different types of radiation. This app was produced by Dr. Richard Barker, and the work was supported by grants called NASA TOAST and NASA Mango. TOAST, Test of Arabidopsis Space Transcriptome. Mango, Meta-Analysis of GeneLab Omics.